This video will demonstrate the steps required to design and order all necessary reagents for creating a single nucleotide change or SNP in a target transcript using the Invitrogen TrueDesign Genome Editor. After logging into Thermo Fisher Connect and accessing TrueDesign Genome Editor, you have several experimental options to choose from. For this workflow, we will choose SNP Edit and click Next. On the next screen, you'll see 5 species to choose from. Select the host organism for your SNP experiment and click Next. You'll then select gene symbol or Entree ID as the input for how you will identify your gene of interest. If you want to target non-transcribed regions, you can use the DNA sequence or chromosome locus option. Click Next. When you begin typing in the gene identifier box, a filtered drop-down list will appear. Select your gene of interest from the list and click Search Gene. The next screen will display all of the protein coding transcripts for your selected gene. Please note that the principal transcript for the gene is displayed at the top and is selected by default. If there is a different transcript you wish to target, select that. You can easily click the gene symbol hyperlink to be taken to the NCBI website where you can visualize the transcript maps. When you have made a selection, click Next to proceed to the sequence editor view. The sequence editor will update to show the entire transcript's topology along the top of the screen with a zoomed-in sequence level view below. TrueDesign Genome Editor also displays genetic variants present across populations in the gray bar above the DNA sequence. Click on the genetic variant nucleotide to view details such as its frequency and genomic position. It is important to pay attention to genetic variants as they can affect the efficiency and specificity of guide RNAs. To view variants in a specific population, click on the drop-down on the right. Use these views to navigate to the region where you will edit your SNP. In the sequence editor, select a single nucleotide as you would in a text document. A selection box will appear prompting you to select the replacement nucleotide. Click OK. The view will update to display an edit list to indicate the nucleotide change and its position. You can undo the change by clicking the blue Undo arrow. The SNP change will also be indicated in the sequence editor. At this point, you also have the option to introduce a second edit within a 30 base pair region of the SNP edit. You can choose a 30 base pair insertion, deletion, or a second SNP edit. For this example, I'll choose a second SNP. When your edit list is complete, click Next. This step might take a few minutes while software searches for the region for available TAM sites and assesses the guide RNA designs for efficiency and specificity. When complete, you will see a table with 2 tabs, CRISPR and TALEN targets. Each row in the CRISPR table displays a CRISPR guide RNA design, its PAM site, its weighted score for efficiency and specificity, the number of predicted off-targets in the reference genome, the edit site distance from the nearest SNP, and the DNA oligo required as a knock-in donor template. If you wish to find off-targets in the variant genome selected on the previous page, click on Search in the Off-Targets in Variant Genome column. This search will take an additional 70 to 90 seconds. Green checkboxes indicate recommended guide RNAs due to score and proximity to the insertion site. Visualize the guides on the sequence editor display by clicking anywhere in the row. Genetic variants are indicated below the guide RNA and PAM sequence to easily identify guides whose activity might be affected. Click on the number in the off-targets in the reference genome column to view more details such as the location, the probability of cutting at the off-target site, and annotations of whether the off-target site is in the exon or intron of a gene. Click on the number in the off-targets in the variant genome column to view more details about the off-target sites in the variant genome as compared to the reference genome. Off-target sites that have higher off-target activity in the variant genome are flagged by an orange warning symbol. To view the TALEN targets, click on the TALEN tab of the table, and similar information will be displayed for each TALEN pair. To select one or more guides or TALENs to add to your experiment, use the checkboxes in the table and click Next. The summary page will display all of the reagents needed for your SNP editing experiment and give you the opportunity to add additional products to complete your workflow. For a SNP edit experiment, the guide RNA, donor DNA, Cas9, transfection reagent, and the GCD or sequencing primers are selected by default. Some of these can be unselected by clicking on the checkbox next to them, or a different quantity may be selected from the drop-down lists. Lastly, you may add additional items such as positive and negative experimental controls. When you have completed your product selections, click on the Download Designs and Protocol to download your entire experimental design for future use before clicking on Add to Cart for easy, one-step ordering of all selected reagents.